In this video, I'll review concepts from Chapter 3 of Human Anatomy and Physiology, 11th edition. This chapter is focused on cells. In particular, we'll be looking at membrane transport, or the movement of substances from or between the inside and the outside of the cell. There are two major methods for moving substances across the plasma membrane. We have active transport and passive transport. And the difference between these two is whether or not they require energy. Active transport requires energy to get done, and usually that energy is in the form of ATP. Passive transport, on the other hand, does not require energy. So let's start with passive transport. There are three classes of passive transport. We have simple diffusion, facilitated diffusion, and osmosis. All three of these classes are passive because they all are movements of molecules down their concentration gradients. So what down the gradient means is from high concentration to low. So molecules will passively flow from high to low down their concentration gradient. This means that no energy is required. There's some general rules of diffusion. Diffusion is basically just random movement of molecules. And with random movement of molecules, you're going to see them move from areas of high concentration to lower until everything evens out. So it'll eventually, within this closed container shown in the illustration, eventually we get an even distribution. But it starts with a high concentration in the center. So it's this sort of natural movement of molecules. Energy, again, not required. And the speed of diffusion is going to be determined by three big factors. So molecule size, so bigger molecule means slower diffusion. Temperature, so as the temperature gets colder, diffusion is going to slow down. And distance, so it's going to take a longer time for the molecules to diffuse further. So the further the distance, the slower the diffusion. So now let's look at one of those three types of passive transport, and that's simple diffusion. So as the name implies, this is the simplest of these three. This just means that it's going to be a non-polar, lipid-soluble substance. So this basically means it has to be very, very tiny or hydrophobic. Basically, what this is talking about is it has to be able to pass through the hydrophobic portion of the lipid bilayer. So a molecule that can do that, something that is sufficiently small or hydrophobic, can just pass right through. And that's simple diffusion. So if something is high concentration outside, low concentration inside, as shown here, you're going to see a net diffusion into the cell. Some examples of things that are going to travel across the membrane by simple diffusion include oxygen, carbon dioxide, as well as some fat-soluble vitamins. We also have something called facilitated diffusion. This means that there are certain molecules that can be transported passively down their concentration gradient. So shown here, we've got some sort of solute that is going from high to low. However, they're going to require some sort of carrier or channel protein to get across. So examples of this would be solutes like glucose, amino acids, and ions. So things that are large and or have ionic charges. So in those cases, they can't diffuse across this lipid bilayer. They can't get past the hydrophobic portion. So instead, we have transport proteins that are going to allow them to still diffuse down their concentration gradient, but in this case, we call it facilitated because the proteins are facilitating the process. Now, finally, we have osmosis. So this is sort of a special case. It's still diffusion, but you can see here there's actually a little bit of simple diffusion going on and a little bit of facilitated diffusion. What makes osmosis special is it's talking specifically about the diffusion of water. So remember, water is the major solvent of our bodies. And so we need water to be well balanced inside and outside. This is going to determine the concentration of solutes in all the different portions of our body. One thing to note is that even though we have this hydrophobic portion of the lipid bilayer, water is small enough that some molecules can sneak past. So again, they can travel by simple diffusion in this case. However, if you really want to speed this up, we also have these transport proteins called aquaporins. And you'll see these come up in a few different places, but in particular, you'll learn about them in the kidneys. So these aquaporins are proteins that are going to allow for much faster transport of water down its concentration gradient. Now, osmosis is a little bit strange in that, remember, water is the solvent, so it's the thing that is going to define the concentration of other molecules. So when you're thinking about osmosis, for me, the easiest way to think about this is that water will move from the side of the membrane with low osmolarity to the side with high osmolarity. So that means that water is going to move toward the side of a membrane with more concentrated solute. So here's a definition for you. I use the word osmolarity here. Osmolarity is the concentration of solute. The units are in molar, so molarity, molar is the unit. And in living organisms, that solvent is going to be water. And the solute, so that solute that we're talking about here is substances dissolved in the water. So if you can imagine, you've got two different fluids. One has water and very little substance dissolved in it. One has water and a lot of substance dissolved in it. Now, the overall volume in these two cases is equal. However, in the example that has more substances, higher substances, so in this case, 10 molar is higher than 1 molar, this has more of that volume being taken up by substances and less being taken up by water. So water is going to move down its concentration gradient to where there's less water. And in this case, that is outside. So that's all very complicated. Again, the way that I like to remember this is that water moves toward higher concentration. So water in this case is going to move from 1 toward 10. So it will always move toward the higher number. This is an example showing intracellular fluid at 1 molar and extracellular fluid at 10 molar, for example. So in this case, water is going to flow out of the cell until these two are equal, or really until the cell basically runs out of water. In order for osmosis to work, that membrane needs to be selectively permeable. What that basically means is that it's going to allow water to pass back and forth between the inside and the outside of the cell, but it's not going to allow those solutes to move back and forth. So the solutes stay the same, and the water has to move. I'll remind you again, the reason for that is that osmosis is based on differences in solute concentration. So if solutes can just freely move, then those solutes will diffuse back and forth until they're equal on their own. This becomes very important, especially in animal cells that lack a cell wall that helps stabilize them, because of this thing called tonicity. So when we see a red blood cell, what we really want to see is a cell that looks like kind of like a donut, or like an inner tube that you might ride, say, down a river. So in this case, these are red blood cells that are existing within an isotonic solution. So your blood plasma needs to be isotonic. What that really means is that the concentration in the extracellular fluid needs to equal the concentration of substrate or of substance in the intracellular fluid. When that's true, the cell volume is going to be stable, and your cells will look like this nice donut shape that can fit very neatly through your little tiny capillaries to all the tissues of your body. If you were to place that blood cell into a hypertonic solution, what that would mean is that the cell volume is going to shrink. So that's what's shown here. This cell has been placed into a hypertonic solution, and that has caused the cell to shrivel. And that's because the substance concentration in extracellular fluid is greater than intracellular fluid. So this hypertonic solution is the same as the example I used in this previous slide. So this would be a hypertonic solution, 10 molar versus 1 molar intracellular. So you can see here, hypertonic solution causes a cell to shrink. And finally, we have a hypotonic solution. So this is an example where the extracellular fluid concentration is lower than the intracellular concentration. And when you put a cell into a hypotonic solution, you'll see that it's going to actually enlarge. So it's going to take all that water, and the water is going to move by osmosis into the cell. And the cell will blow up and eventually burst. So these changes in shape can drastically affect a cell's function. So for instance, when this red blood cell shrinks or shrivels, it is no longer going to be as capable of binding and releasing oxygen. Additionally, it's no longer going to be as capable of moving through capillaries, and it might get caught on capillaries, or it might actually get caught on other cells. When it's blown up, again, can't fit through all those capillaries as it's moving through the body. And eventually, it might burst, and in that case, it's no longer an intact cell. So these changes in shape can be very important. So what we really want is an isotonic solution for our cells. So now, that was passive transport. So we talked about three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. Now we're going to talk about two classes of active transport. So those include what's called active transport, as well as something called vesicular transport. So for these two classes of active transport, molecules can now be moved against their concentration gradient. So that means that you can take a molecule and move it from a side of the membrane that has a low concentration. You can actually move it to the side that has a higher concentration. This means that it's going to require energy. So this is going to allow you to move even larger molecules. Additionally, it allows you to move polar molecules, and it allows you, most importantly, to move them against their concentration gradients. So here's an example. So active transport, that first type of active transport, and in fact, this is also called primary active transport. This is going to require carrier proteins. Often these are called pumps. So they are going to bind specifically and reversibly with substances that are being moved. So what's shown here is a sodium-potassium pump. So it's going to be able to bind to both sodium and potassium and move both of those molecules against their concentration gradients, so from low concentration to high. Because this is going really against physics, this means that you have to put energy into these pumps. So in this case, this pump is an ATPase, meaning it is going to hydrolyze ATP. It's going to use the energy from that phosphate bond in ATP to power the pumping of these ions from one side of the membrane to the other. So the sodium-potassium pump, the reason it's chosen for this example is it's actually the most studied example of active transport. It is vital to the function of all of your cells. It moves sodium out of the cell, and it moves potassium into the cell. Both ions are being moved against their concentration gradient, so you're building up a lot of potassium inside the cell, and you're building up a lot of sodium outside the cell, and that's why it requires ATP. The reason that it's found in all of your cells is that it's vital for maintaining what's called a resting membrane potential. So I'll talk about what is a membrane potential. So this is something vital for cell function. It's a voltage. So it's a difference, a separation of ions. So you'll have maybe more positive ions outside the cell and more negative ions inside the cell, and that's going to be what creates the membrane potential. It's especially important for what we call electrical cells, and these are nerve and muscle cells. They're going to use those membrane potentials and changes in membrane potentials to trigger signals and to trigger contractions. So in addition to that primary active transport, we also have vesicular transport. So this is going to involve transport of large particles and fluids across the membrane. And as the name implies, the defining feature here is that it's going to use vesicles. So these are membranous sacs. So here we see some endocytosis happening. This is transport of substances into the cell. So we have substances coming from the outside. They're getting taken up here by this vesicle and moved into the cell. So that movement into the cell in vesicles is called endocytosis. On the bottom, you can see the opposite is exocytosis. So exo means out. And in this case, those substances that were moved into the cell previously are now being released into the outside via these vesicles. So it's transport out of the cell by vesicles. We can break endocytosis, that movement of substances into the cells in vesicles, into three different types. So we have phagocytosis. This is endocytosis of large molecules or even microorganisms. I think that's what's being shown here, or some sort of large particle is being shown here. We have pinocytosis. This is endocytosis of fluids with very small solutes. So phago is large, pino is small, but both of these are bringing substances into the cell in vesicles. And then finally, we have receptor-mediated endocytosis. And this is going to be substances are going to bind to specific receptors, and then those receptors are going to trigger the import of those substances into the cell. And so what makes this different from these other types is that these are going to be very specific substances binding to specific receptors that only respond to those substances. Whereas phagocytosis and pinocytosis are going to respond to a much broader range of substances. Now we can look at the opposite, exocytosis, movement out of the cell. There are four major steps. We have first, that vesicle is going to move toward the plasma membrane. Then through some steps that we won't get into in this class, it is going to fuse with that membrane. So the vesicle fuses with the membrane, shown down here. Next up, a pore is going to open. So you can see here that that pore is now opening so that the substances within the vesicle are now open to the outside. And then the final step is that those substances are actually released to the other side of the membrane, to the extracellular space. So those are the four steps of exocytosis. And I'll leave you with a question about membrane transport. The maintenance of the resting membrane potential by the sodium-potassium pump is an example of, and I've listed here 4 different types of membrane transport.